Hello, everyone, and thank you for joining me today. My name is Yash Dixit, and I am a scientist in the meat quality team at AgResearch. My research background is hyperspectral imaging and diffuse reflectance spectroscopy for food quality and safety, majorly meat. Let's start with sharing my presentation. In the current industrial scenario, meat industry is looking for non-invasive spectroscopy-based measurements or solutions for real-time quality monitoring systems, and our research contribute to that particular interest. The topic of my presentation today is a global calibration model for red meat quality. When I talk about calibration model, it is a model based on hyperspectral imaging data. And with regard to red meat quality, I'll be focusing on two of the most important quality indicators in red meat, that is intramuscular fat and pH. Let's start with the overview of my presentation. I'll give a quick overview of meat and light interaction, a quick discussion around hyperspectral imaging, the model building process, why do we need a global model, the study, and finally the conclusions. When the light is incident onto the meat, there are few different phenomena which take place. A regular reflection, commonly referred as specular reflection or mirror-like reflection, where there's little or no interaction with the sample. Then there's external diffuse reflectance, where there is interaction with the sample surface and which is important to us. Then there a part of light which is absorbed, a part of light which is transmitted, and a part of light which is scattered. In terms of external diffuse reflectance, there's various factors which affect these measurements. That is your meat type, your muscle type, fiber direction, also animal related factors such as gender. Hyperspectral imaging. Let's quickly talk about it. Hyperspectral imaging is a combination of imaging and spectroscopy. There are few different hyperspectral imaging systems. The most common one which you will come across is the line scan system. As the name suggests, the image is scanned by one line at a by scanning one line at a time, either by moving the sample here in this image or by moving the camera. And there are snapshot camera systems which works very similar to your camera phones, and there are point scanning system as well, but we won't go into the detail. What kind of data which we get from the hyperspectral imaging? What we get is a 3D array known as hypercube. It is a stack of 2D images uh, at different wavelengths. These wavelengths depend upon the wavelength range of your hyperspectral imaging systems. Here we are will be focusing on the visible and IR region. As you can see from this image, for each pixel of your hypercube, you get a spectra. The spectra gives you information about particular composition. Also, it is somehow related to your structure as well. But these bands are related to your particular composition attribute, for example, fat or protein or moisture. Once you get that hypercube, the first step is to remove the background or extract your region of interest. In order to do that, we use image segmentation. And once you have the processed hypercube, it is generally averaged to get a single average spectrum. Now, how it is better than a single point spectroscopy? In a single point spectroscopy, you'll collect spectra at few different points, but in this case, you are covering the whole sample, thus overcoming the heterogeneity of the sample and giving better representation of your sample. Once you have the um, average spectra for all the samples in your particular experiment, the next step is for spectral preprocessing to overcome any nonlinearities due to various phenomena which I mentioned in the previous slides, such as scattering. Also, baseline shifts due to undulations in your sample surface or sample thickness. And finally, the model building using chemometrics. This is how a usual model building process works. Imagine an experiment where you are trying to build a model for a beef strip line sample. You have n beef strip line samples. We collect the hyperspectral imaging data for it, process the data, then you get a process data matrix. This data matrix For in this data matrix, for each row contains the average spectra for the each sample, depending upon the wavelength range of your system, and each column corresponds to each wavelength. Also, collect the reference data, in our case IMF, FAT and pH, and then you have reference data matrix. Then using these data matrix, we develop a model. Now this particular model would be valid or will work for similar kind of sample in a similar environment. That is beef strip line samples in a similar environment. If it was done in lab, it will work in lab, or in a pilot plant, it will work in pilot plant. But what if you have different meat samples or meat species, different muscle types, samples collected in different slaughter seasons, your experiments being done in different environment, for example lab or a processing plant. You have you have used different parameters for your system depending upon your sample type, different people using it. Is there a possibility of developing a global calibration model? And this forms the basis of our research. A novel approach to develop global calibration models for predicting IMF and pH across different meat species and muscle type. We have used three different red meat species, that is beef, lamb, and venison, and 20 different muscle types. Initially, for data collection, collected data from 8 different experiments or trials, 2 for beef, 5 for lamb, and 1 for venison. And across them, there were 20 different muscle types. Collected the reference data for fat and pH and hyperspectral imaging data using a line scan system, which worked in the wavelength range of 550 to 1700 nanometers. So in total, we had 8 different data sets. And these are details of each experiment. As you can see, there were different number of samples in each uh, experiment. There are different types of meats being used, and these experiments were done from the year between the year 2017 and 2019, and in different locations. That is, some of them were done in lab, some of them were done in pilot plant. Also, you'll observe that there's certain differences in the camera parameters depending upon the samples being measured, different people using that instrument or doing those trials. Let's visualize the reference data. It is important to have a good range of your attributes which you're trying to predict, for example, IMF and pH in this case. Otherwise, your uh, model would be it would be difficult to develop a good model. It would be uh, very tight fitted or uh, underfitted, overfitted model. So if you look at this box plot on the left hand side, uh, which is about IMF percentage, there's a good range of good spread for beef, tighter range for lamb, and very tight for venison. But all of them together are giving a good range for IMF. Similarly, for pH, you see a good spread for beef, tighter range for lamb, and very tight range for venison. So in case of both attributes, uh, when we collect all the data, you are getting a very good range of IMF and pH, which is important to develop a good calibration model. Now let's talk about the data analysis or the model building process. So we collated all 8 different data sets from the experiments into a single data set. Then we split the data into 70% calibration, 30% validation data, and we made sure that both these data sets contain meat from different species and different muscle types. For model development, the calibration data set was used. Initially, we used the strategy of repeated double cross validation for selecting optimum number of latent variables or components for the model, we built the partially square regression model, which is a modeling technique for which is a predictive modeling technique. So repeated double cross validation is a robust method of selecting the optimum number of components or latent variables for your model. Then once these optimum number of components were obtained, we used k-fold cross-validation strategy using 10 folds to cross-validate our models. Then finally, these models were externally validated using the validation dataset, which is unseen by the model itself. So we developed 2 models, one for IMF and one for pH. And these are the results for that models. As you can see, we used few different pre-processing techniques. the most that is standard normal variate transformation, which is the most common one, along with multiplicative scattering correction and first derivative as well as second derivative. And as you can see, the best results were obtained for SNV. If you look at the calibration part, and here there are squares or coefficient determination cross validation, which were quite high, 0.85 and 0.815 mf and pH. Also, the standards of cross validation were quite low, 0.86 and 0.15 for mf and pH respectively, which shows a good fit for the model. If you look at the prediction performance of the model, excuse me, model here, a high R squares were obtained for prediction as well, 0.88 and 0.86 for MF and pH, and low standard errors of prediction. If you look at the slope and intercept, the slope is nearing zero. Slope is nearing 1 and intercept is nearing 0, showing the so it's following the linear equation. Also, there was negligible bias and high RPD as well. Now let us visualize the predictions. So these are the prediction plot. So this is the prediction plot for IMF. If you look at this prediction plot on the left hand side, which is color coded based on the meat species, you'll see there's a good spread for beef, as we saw from the reference data as well. A tighter range for lamb and venison. So here you see that irrespective of the meat species being used, the models were able to predict IMF quite well, as seen from the table or statistical indices as well. Also, if you look at the right hand side plot, which is a similar plot color coded based on muscle type, you'll observe that most of the muscles were longissimus, but there were other muscles as well. So irrespective of the meat type and muscle type, the models were able to predict the IMF quite well. Similarly, for pH, a good range for beef, a tighter range for lamb, and very tight range for venison. And here also, in terms of muscle types, also you will see that irrespective of the muscle type and meat type, the models were able to predict the pH quite well. Now, VIP scores plot is a valuable method of understanding and visualizing the importance of various variables in explaining the y variance. Here our variables are our wavelengths and y variance is IMF and pH. If you look at this top plot for IMF, and here if you look at this range from 1100 1400 nanometers, these are bands related to CH second overtone, uh, which are related to radical CH, CH2 and CH3 of triglycerides, which is the building block of fat. So this portion is related to fat itself. Also, if you look at this peak around this is around 927 nanometers, related to CH third overtone, also related to fat, which tells us that the models were able to capture the fundamental properties or fundamental bands related to IMF. In terms of pH in the diffuse reflectance spectroscopy measurements, it is a combination of bands and visible and NR region. pH is related to your meat color and meat's water holding capacity. Darker the meat, higher the pH. Higher the pH, lower the water holding capacity of meat. If you look at these peaks around 580 and 600 nanometers related to oxymyoglobin and myoglobin, uh, aiding to the color of your meat and helping in prediction of pH. Also, this area around 980 nanometers related to OH stretching, uh, OH stretching also aiding in prediction of pH. So in both these, from both these plots, you can see that the models were able to capture the fundamental properties or fundamental bands related to IMF and pH, illustrating the global nature of these models. In order to further investigate, we compared the VIP plot for IMF to one of our previous study. If you look at uh, uh, this region of VIP scores plot from 900 to 1700 nanometers and compare it to one of our previous studies, you see the shape is very similar, which tells us that the model was able to capture the uh, fundamental properties related to fat irrespective of difference in experiments. The interesting thing here is this particular experiment was done using a far more cheaper device called a mini NIR spectrophotometer, which is almost 20 to 30 times cheaper than this. And also, the meat matrix was different. So, in the current experiment, we used intact meat, while in the previous experiment, it was freeze dried lamb meat powder. So, from this particular comparison, there are few conclusions which we can make. Firstly, uh, the current global calibration models was able to capture the fundamental uh, bands related to IMF, irrespective of the instrument being used and irrespective of the meat matrix, which illustrates its global nature. Also, there is the potential of using a far more cheaper device to develop such global calibration models, which I believe will be more appealing to the industry. Now, finally, the conclusions. As we saw from the model statistics and the plots, the global models were able to describe several factors such as chemical composition of your meat, a meat structure, that is muscle type, and meat species as well. Models were able to capture fundamental bands or peaks related to IMF and pH as seen from the VIP plots. Also, when the VIP plot of IMF was compared to a previous study, we also um, which also showed that model were able to capture the same information in respect to the sample metrics. Also, that comparison showed us the potential of using cheaper instruments for developing these kind of models. Finally, in the digital era, as I also mentioned initially during the presentation, these kind of models can be part of automatic quality monitoring system in a meat processing plant, where where they want to predict intermuscular fat and pH and across different red meat species and across different muscle types. Finally, the acknowledgements. I would like to thank the funders, MBI New Zealand, and AgResearch Strategic Science Investment Fund. A big thank you to our industry collaborators. And last but not the least, to thank you to all the team members for their individual contributions. Thank you.